So the first thing that I really want to point out is sometimes we look at things that are in our homes and we don't really realize what they have in common. They might seem obviously quite different. Some go on cars, some we eat, some we consume for other reasons, but major things that most everything that we interact with or have in our homes have in common is that they have the addition of synthetic chemicals. So I put a couple as an example here. So this one right here is called 6-PPD. It's a coating on tires that has actually had a lot of issues for fish recently. This one up here is S-metallochlor, so it's another type of pesticide that is being put on corn and on soy to avoid the growth of other things that we don't want in our croplands. And right here, this is EE2, which is the main component in birth control. So these are all things that we interact with, and we find ways to use them without necessarily having them harm us, but that's for people. And a lot of other organisms like those that live in waterways are not people. So in this case, when we have these chemicals in our homes, where do they eventually go? And as I'm sure you're aware, through one way or another, they eventually make their ways into our waterways. And that can affect everything that grows in there, whether it's algae, whether it's fish, whether it's invertebrates, anything like that can be affected. So the traditional way of doing toxicity testing is you take one chemical at a time and you expose different types of organisms to it, and you try to see how they react to it. And then you determine acceptable environmental thresholds based on those reactions. The main problem is that you're setting these environmental regulations for each chemical separately. And as I'm sure you're aware, there's not just one chemical in any one place at any one given time. There's, most of the time, hundreds or thousands of them. These are a few that I've decided to put up, but I can guarantee that there's many, many more than these. So, with all of these chemicals interacting together, the main question is, well, how does this combination affect the overall toxicity? And in this case, how does it affect wild fish? So that is what my research looks at. And I'm trying to see, does the setting of individual thresholds for each chemical, does that still protect fish? Or when you have this combination acting together, does it no longer protect fish? Is it actually too high? So as far as toxicity testing goes, traditionally, it's quite a lengthy process. So usually we use adult fish, and it can take an extended period of time. It can take months or years to do full toxicity testing. And because of that, as of right now, with the methods that we currently have, we have about a 10-year backlog on testing chemicals extensively. So a lot of them have been approved, but they have to undergo more extensive testing, and that has not been done yet. So what we're trying to do is we're trying to change the methods to a lot of new approaches. So instead of using adult fish, a lot of these new approaches use hatchlings, like larvae or juveniles. So it takes a lot less time to bring them up to that life stage. It's a lot faster. And because it's a lot faster, and it's also easier to do multiple chemicals at a time, it's a lot less expensive. So as I said, instead of taking months or years, it can take anywhere from hours to days. My exposures are about 24 hours long, which is way shorter than the average of weeks or months or years. The other thing is that there's also been a shift towards, instead of using what we call organismal effects, so is there an impact on reproduction? Is there an impact on physical deformities? Is there an impact on fish are less likely to survive? We look at transcriptomic markers, so that's gene expression levels. So for my test subjects, as we said, wild and laboratory fishes. So in this case, the zebrafish right here is my laboratory species, and then copper red horse here is actually a species that's found in the Richelieu River, and is currently listed as endangered. So we're trying to find reasons as to why that is the case. We currently don't know that much about the species, but there's been an ongoing effort for about the last 20 years to bring their population levels back up. And understanding the genetics behind how they interact, or how they respond rather, to certain chemicals that are found in their waterways can actually really help that. Another reason why we use early life stage fish is because of the difference in size. So this gives you kind of an idea of the difference in size of the fish that I work with. So zebrafish, when they're fully grown, are a few centimeters long, maximum. Copper red horse can get up to 30 pounds. So when they're in their early life stages, there's only about a centimeter difference between them size-wise instead of, massive differences. So this can make it a lot easier to have a lot of fish all together and do the exposures all at once, because the amount of space that it would take to have, dozens of fish that are 30 pounds is huge compared to the amount of space that's needed for fish that are, 1.5 centimeters long. So now for my experimental setup, what I'm doing is that I'm comparing exposures of fish to a specific chemical within different contexts. So one of that is lab water. That's clean water, no contaminants, no nothing. And I look at what the gene expression shows me. Like, at what concentration are they being affected by these chemicals? What kind of effect that's having, which genes, things like that. Then what I do is that I put them into different water types. So one of those is river water, and we do a couple of collections, one of which is early in the season when there's not a lot of pesticides. After that, it's later on when there's a lot more spraying and there's a lot more runoff. So if we take the example of the river water, then we redo the exposure, same chemical, same fish, same stage of life, and we look at their genetic expression analysis, and we see, is there an impact? Are they more impacted? Are they impacted at lower concentrations? And then we compare to see if the background contamination has an effect on the toxicity of this chemical. And if they're impacted at lower concentrations, or they're impacted, the genes are impacted more, or there's more genes that are impacted, their expression levels, then that can tell you that there's something in the river water that's contributing to the toxicity of that chemical. So I'm just going to give a quick overview of what my exposure process looks like. So first of all, because we work with fish that are freshly hatched, we actually have to hatch them ourselves. So we get the eggs, we bring them in, we put them in aquaria with bubblers, and then we let them develop and hatch over time. Once they're hatched and we get the larvae, what we do is we call it a microplate-based assay. So we put the fish into each well of a microplate, in this case a 24 well plate, and then we add the chemical and whatever water type that we're using into each of the wells. And what it looks like is that there's actually not just one concentration, we do 12. So we go from a control well all the way on the left side here, and it's increasing concentrations all the way to very high concentrations, which we expect to cause mortality. This gives us a full range of effect. We go from expected no effect, like very low concentrations or controls, all the way to very high concentrations, and it gives us a smooth gradient of impact. So we're able to actually see the increase of impacts. Then what we do is that we pull them, we pull the larvae, like put a couple of them into the same tubes, then we freeze them, and then we use them for transcriptomic analyses. So for each chemical, I have to do this 3 times with each of the 3 water types that I just talked about earlier. Then I don't only have 1 chemical, I have 3. So, and this is for 1 species of fish, do it twice. It's hard to put it into context just by speaking about it, but for my master's thesis, I have to do this about 18 times. So this is a lot of manpower, it's a lot of work, but compared to what would have to be the case if you use, adult fish or over longer periods of time, this could be, multiple PhDs. So these are really methods that are allowing us to do a lot of important work a lot more quickly, and it's allowing us to get some results of products that are currently being used a lot more quickly, so that we can make recommendations for new limits if the current limits aren't appropriate. So this is really important for not only human and animal health, but also just the overall sustainability of agriculture and industry, since most chemicals in the environment come from these sources. It's not necessarily all me and you, it's a lot of farming, it's a lot of industry. So making sure that these limits are appropriate is really important to make sure that our waterways aren't irreversibly harmed long term, and to try to keep those limits down so that we're able to enjoy the waterways, both us as people and also all of the other animals that rely on these waterways. So thank you for listening to my presentation. This is my lab. If you want any more information, you can always scan the QR code and visit the website and see what other people are doing in the lab.